Okay, so let us catch up from where we left. Let's start with the definition of genotype and phenotype. So in computer parlance, genotype will be the back end and the phenotype will be the front end. Meaning, genotype is the actual sequence of the DNA which dictates how the phenotype will be. Phenotype is everything that is visible, okay, or testable. And phenotype is dependent on what the genotype is. You will often hear terms of wild type versus mutant. I'll share a funny story with you without naming names. So in the early days in 2007, when I applied for a grant from one of the funding agencies in India, premier funding agencies in India, I talked about doing a mouse genetics experiment with wild type versus mutant. And one of the reviewers thought that by wild type I mean wild. So the question was, how would you get mouse from the wild? So let me be very clear here. By wild type, we mean that it is a strain of mouse which is, again, a standard. Okay, it is a laboratory standard defined. So it has a known DNA sequence and its characteristics are known. These are being used for more than 70, 80 years now in the lab, in different labs, of course. And the same mouse lines are propagated through generations and are used by different investigators throughout the world. So whenever you hear the term wild type, whether it is mouse or chick or fly or any organism or yeast, bacteria, it is a particular genotype phenotype combination that has been defined as the standard. Okay, so that is wild type. And any variant from that, genetic variant from that, is a mutant. This has nothing to do with wild. Wild is what you get out in the jungle, in the river, in the food waste, etc. Those are not laboratory bred. And the reason those are not used is because there is a good possibility that there will be genetic variations from one wild species to another wild, like one set of wild organisms to another set of wild organisms within the same species. So there will be naturally occurring genetic variation, which will then make it difficult to relate genotype to phenotype. So in wild type, what we have is, whatever the organism is, because I work primarily with mouse, so it is easier for me to give you examples of mouse, is that a wild type male and wild type female is mated. Then the pups that will be born, those are brothers, there will be some males, some females, so there will be brothers and sisters. And then you mate brothers and sisters, and then you get the progeny. That way, you do not allow any opportunity for genetic variation to come in. It is the same two alleles from the original stock of the mother and the father that is being propagated in all these strains, even now, after 70, 80 years. So, because they are maintained through this brother-sister mating, therefore, there is no genetic variation. Now, in that kind of a background, if you make a genetic change yourself as part of your experiment, you know which gene you are changing. And then you can observe the phenotype, how it is changing. And then you can say, oh, so this is the gene that is responsible for this particular function, meaning, think of the switchboard analogy in the last lecture. If I turn off a gene in a wild type mouse and I see that it is no longer black colored, but it is white colored. So you know that the black color comes from that gene that I turned off. Or I introduce a new gene or a gene at a higher, like, which will be produced in more quantity than normal. And you see that the mouse suddenly has bigger body mass. So you know that, okay, this is somehow related to body mass. Okay, there are some fantastic experiments done where a gene called double carotene was introduced. So it's a gain of function mutation in mouse, and the mouse brain size doubled. Okay, so this is how we do experiments in genetics, and that's the reason why we need a wild type mouse whose genome is absolutely defined, and in a given set of animals in a particular laboratory, in a particular strain, the genotype is identical between any two individuals. Okay. Another set of terms that you will hear is haploid, diploid, aneuploid, polyploid, etc. So we will refer to it a little later again. So if you think of the total genetic content, like, like in our cells, we have 22 pairs of autosomes, and if you are a male, 1 X and 1 Y. If you are a female, 2 X's. Now, this, if we eliminate the pair part, 22 autosomes and either an X or an Y, that is the minimum set of the genome. Okay, so that is one copy of each gene in the set. That is the haploid set. Okay, so haploid is 1. If it is 2 copies of each gene, then that is the diploid set, which all our cells, other than the germ cells, are. Okay, so in a diploid cell, because I am a male, so I will have 22 pairs and an X and a Y. Aneuploid is, in a normal organism, just one chromosome will be missing. Okay, that is aneuploid. Okay, in humans you do not get to see those at all because they don't survive. But in certain organisms you can genetically engineer it, and they exist. So those are aneuploid. And polyploid is where you have an extra chromosome. So this is different than misexpression of a gene. Like, say, the gene for glucose metabolism, hexokinase. Okay, you will certainly have one copy of that. If I introduce in a mouse an additional copy of hexokinase, that will not be called a polyploid mouse. That will be called a hexokinase gain of function mutant mouse, or a hexokinase overexpression mouse. That's all. But that is not a polyploid. A polyploid will have the whole chromosome extra. Okay, and these all have their own usage in genetics experiment. We will come to that if and when we are there. And then homozygous and heterozygous. Homozygous is that for each gene in both the pairs, in both members of the same pair, the sequence is identical. Those are homozygous. Okay, like let's think of the hexokinase again. So if the hexokinase gene sequence is identical in both the chromosomes, then it is homozygous for hexokinase. It is homozygous for a particular gene always. Okay, if not, which most of us, I would say that almost none of us are homozygous for any of the genes. We are heterozygous because we got one hexokinase from father and one hexokinase from mother, and they are, even though phenotypically they may not be different, functionally they may not be different, but chances are that sequence-wise they are different. So we are therefore heterozygous for any given gene. What is a dominant and a recessive mutant allele? So dominant or recessive is always referred to in the context of an allele. Okay, so the concept is that if, irrespective of the presence of the other allele, if the characteristics are displayed because of one particular allele, then that is the dominant allele. So there are two different alleles. One of the alleles is dominating the outcome. Okay, that's why it is a dominant allele. So let me repeat again. If there are two different alleles, one of the alleles, if that dominates, what the outcome will be? Let's say, like, let's take an example that two alleles in one, gives rise to larger body size. The other one gives rise to normal body size or even smaller body size. These are two alleles of the same gene. But in the heterozygous, if you see that the body size is larger than normal, then you know that it is that one allele which generally gives rise to larger body size that is dominating the phenotype. Recessive allele, its phenotype will be visible if both the alleles are the same. So it is a homozygous. Only in the homozygous condition you get to see the phenotype of the recessive allele. Okay. Haploinsufficient, this is a pure dosage issue. Okay, like the proteins that you have in your cells, they have a function, right? And because end of the day it is pure chemistry, so they will have, like the dissociation constant, they will have a rate of reaction, they will have rate of diffusion, so many physicochemical parameters are there on any of the proteins. Now, because of that, you may need protein produced from both the chromosomes. So we have, for every gene, we have two copies, right? For majority of the genes, the second copy is redundant, meaning if you don't, even if you don't have it, you will not know and there will be no phenotype seen. But there are certain genes. One of the genes that I work with a lot is a transcription factor called SOX9. This is a transcription factor. It is involved in so many different processes. But SOX9 heterozygous, meaning if you knock out SOX9 in a mouse, so one SOX9 is not there, just one copy is there, those mice cannot survive. Okay, so haploinsufficient. Okay, so SOX9 is the technical way of saying this is SOX9 is a haploinsufficient locus, meaning you need both the copies of SOX9 for normal phenotype. Okay. And dominant negative is that, like it is just another dominant phenotype. It will block the process even if a normal good copy of the gene is present in your cell. Okay, so I would request that all of you please go through all these definitions from like working definitions, and do your Google search, etc., to make sure that you know what you are talking about. It is very important that you have very clear concept about all of these terms. Okay. Now let us move one step further. That how do you take any attempt of correlating genotype and phenotype? Okay, so in the middle you see that the cloned gene DNA sequence is right in the center, because that is the immediate objective in all kinds of molecular genetic studies. Okay. Now you can get to the cloned gene by variety of methods. Okay. One method is that like it is just an example. Do not take it as word of God. It is not practical. I'm just giving it to you as an example and nothing more than that. Okay? So you all know about firefly, right? So firefly or jugnum, they have this light at their rear end, right? Now imagine that you found a firefly, which does not have that light in the rear end. So it's a mutant, right? Naturally occurring mutant, which does not have that light. So you know that the gene or genes that are responsible for that light in the rear end, it's phosphorescence, I think. Those genes are not functional in this mutant firefly, right? So if you have a way of covering the genome of the wild type and the mutant, you get to know what are the sequence changes in the mutant. That is a good way of approaching it. It is a very modern way. This was not available 10 years back. Today it is there. But the problem with that will be that, unless it's a laboratory bred strain, there might be so many variations in sequence. Okay? So you will not be able to pinpoint. So we do what is called screening of DNA library. Just to give you an example, say you have an yeast strain. Okay, normal yeast strains, they can use galactose as a carbon source. Okay? Glucose is of course the first preference carbon source for everybody, but yeast, etc., they can also use galactose. So if you grow a cell on galactose containing medium, they will turn on the genes that are necessary for galactose metabolism and they will live happily. But you find a yeast that can grow happily in presence of glucose, the normal sugar source, but not in presence of galactose. So you know that something is wrong. Some gene is not there. So you do not know which gene it is. So if there is a way for you to supplement each gene one at a time, so there are approximately 10,000 genes. So if, theoretically speaking, you have an yeast cell, imagine that, like a cassette, like a cassette player, and you insert one cassette at a time. You play, if it is not the right song, you take it out, play the next one, doesn't work out, take it out, like that. So if you have a way of introducing each of the 10,000 genes one at a time in the mutant that cannot grow in presence of galactose, there will be one gene, right, which will allow it to grow even in the absence of galactose, which will allow it to grow in presence of galactose, which will allow you to utilize galactose as a carbon source. And only that one will survive on a galactose containing medium, right? So then you know that which gene you have inserted. So then you got, okay, so this is the gene responsible for yeast cells to grow in presence of galactose. That is what we mean by screening library. It is just like, say you have thousands of cassettes, they are not labeled. It's important to remember that, they are not labeled. And you want to find out that one particular favorite song of yours, how would you do it? You will have the tape recorder, you will put it in, play it. If it is there, you are happy. If it is not, you play the next cassette and you keep on doing that till you get that cassette where your song will be played. So you screen through your cassettes. Similarly here, you screen through the DNA library. How the libraries are made, how the screens are done, etc. These are very sophisticated experimental techniques. I will talk about it a little later in a future class. Once you know the gene that can rescue growth in galactose, you can take that gene and express it, in E. coli or yeast or mammalian cells, whatever you want. Depending on the requirement, you can express it in any host. And then you will get the protein that is present there. There are ways of knowing which particular protein you expressed. And then you can do the sequencing, you can have an antibody against the protein to figure out where in the cell it is located. And lots of studies can be done. That way you know everything about the gene product, as much as today's technology allows us to do. Sometimes it happens the other way around. Like if you take example of glucose metabolism, biochemists, they figured out that there is an enzyme that adds the phosphate group to glucose and converts glucose to glucose 1-phosphate or glucose to glucose 6-phosphate, and they call it the hexokinase, right? That was discovered by biochemists long time back, much before DNA, molecular biology, etc. became buzzwords of modern biology. But they did not know what is the sequence of this gene. Okay, so there are ways of going from the protein to the gene. Like, one way is that you have a pure protein, you can actually sequence the protein, right? The Edwin degradation you have heard of, so you can sequence the protein. Once you know the protein sequence, because you know which are the codons that can make each and every amino acid, so you can have a probabilistic DNA sequence for the protein. Why probabilistic? Because you do not know which of the 6 codons of leucine is used, which of the 4 codons of tryptophan is used. You do not know that. So it can be any one of the 6, any one of the 4. So the probability will be a very large set. There are computer softwares by which you can actually generate all possible DNA sequences that can give rise to a particular protein sequence. Okay, so this is called degenerate set of DNA sequences. Degenerate because these are all equivalent. They all can code the same protein, but their DNA sequences are not identical because of the codon degeneracy. Now you can take the degenerate set and scan the entire genome sequence of the organism and see which one matches. That way you can get there. Okay, but this requires having the genome sequence known a priori. Like you must already know the genome sequence for this kind of a bioinformatics based approach to work. The way we did it during our time, I'm pretty old, right? So I'm talking about 25 years back, 20 years back. If I have a protein, I can make an antibody against it, right? Now with COVID, all of you know about vaccines. Okay? So you take a protein, inject it in an animal, the animal will make antibody against the protein, right? And there are a variety of ways to decorate the antibody so that it can be easily recognized. You can tag it with an enzyme, which will give you a color. You can tag it with a fluorescent molecule, so you can get fluorescence. There are many ways. Now, you will have this DNA, like genomic DNA library or cDNA library that we will talk about later. You can have a very large set of bacteria where each bacterium will harbor one gene or the other. In that set, it is possible that multiple bacteria will harbor the same gene, but it is not possible that the same bacteria will harbor 2 different genes. Okay, that's an assumption. Okay? And it is almost all the time it is true. So if you have that very large set, you know that one or few of the bacteria will be making the protein that you are interested in, for which you already have the antibody, right? So you can break open the bacterial cells on a piece of paper and make sure that every content of the bacterial cell stays on the paper. There is a process called cross-linking. And then you dip it in a solution containing the antibody. Now, antibodies are very specific. So they will only bind to those spots where the particular protein against which the antibody is made is present. So those particular spots will light up. And you know then which bacterial colony made the protein. So you can go back to the original stock of that bacterial colony, isolate the plasmid from there and do the sequencing. That is another way. There are many ways, we will talk about it later. Now, if you started from the protein, you know one function of the protein, okay, because it was isolated using biochemical methods, right? How do you think hexokinase was purified? That someone had glucose in the solution, had radioactive phosphate, added, a mixture of all sorts of proteins, and found that whether glucose 6-phosphate is being produced. Radioactive glucose, look at it, using sophisticated biophysical techniques, and you say, okay, glucose 6-phosphate is made. Now, you started with a mixture of proteins, you use biochemical methods to narrow down to one protein that can make the glucose to glucose 6-phosphate conversion. Please think about it carefully, that this method gives you confidence that this particular protein or enzyme is capable of converting glucose to glucose 6-phosphate. However, because you are doing it in vitro, in a glass test tube, etc., you cannot be sure that the same enzyme, hexokinase, does not do any other function in the cell, right? Because you have not tested for those. We have only tested whether it can convert glucose to glucose 6-phosphate. Nowadays, we know for enzymes, many enzymes, they have functions that have never been associated with them. This phenomenon is called moonlighting. Moonlighting. Okay? So such moonlighting enzymes are really interesting. Like one enzyme that all of you know the name of is called GAPDH, glyceraldehyde phosphate dehydrogenase, key member in glucose metabolism. It is generally used as a loading control in biology experiments because every cell will have it and have it abundantly. Only recently now we know that GAPDH, an enzyme, also can act as a transcription factor in certain settings. This would have never been possible with the in vitro directed study. So how would you do this? When you knock out GAPDH, you suddenly see that certain genes are not being transcribed well. It's a discovery. You chase it and you figure out, this particular protein also acts as a transcription factor. So therefore, the reverse genetics is to discover the entire set of functions of a protein that you knew from the beginning. While forward genetics is you only know the function because in the mutant it is lacking something. And you start from there and get to know everything about the protein. Okay? So these are the two different complementary approaches that are used to connect genotype and phenotype. Okay? Now, before I move to talking about hardcore genetics, let us quickly go through cell cycle, because I want to refer to meiosis once. Okay, so before I talk about meiosis, let us quickly go through mitosis. That you know that DNA is there when it is supposed to be duplicated, then the cell prepares itself for the duplication process, and you call it, the G1 phase, gap 1 phase, and then the actual synthesis happens, which is the S phase, okay? And then, after S phase, you have every chromosome duplicated. And then, eventually it will have to be redistributed into 2 daughter cells. But before that, there is another gap phase or G2 phase. And then in the mitosis phase, during mitosis, and actually the DNA content, now duplicated DNA content at the S phase, now duplicated DNA content is distributed to 2 different daughter cells, right? So, if we are talking about a diploid animal, in the beginning of G1 phase, you have 2N DNA content, right? One from the mother, one from the father. It's a diploid content, so it has 2N, every gene in 2 copies. Here, you have 4N, every gene is there in 4 copies. Between the end, like the end of S phase to the beginning of M phase, like throughout the G2 phase, every DNA is present in 4 copies, okay, 4N. Now, you know that DNA can bind to ethidium bromide, tropidium iodide, etc., which fluoresce, right? So, just an extra information, it has nothing to do with this lecture per se. So, if you treat these cells with tropidium iodide, the total amount of tropidium iodide that will be incorporated in here will be double the amount of tropidium iodide incorporated in here. So this will be more fluorescent than this. You can use this difference in fluorescence to separate the cells that have done their synthesis versus the cells where it is still 2N. Okay, this is different. So here I have elaborated it for you that what is the haploid content, diploid content, and all of that. You look at it later. Now, to understand genetics properly, you need to understand meiosis. Okay. So in meiosis, first we will deal with the simpler thing. Here again, it is 2N content, right? Now it will undergo the first round of meiotic replication, meiosis 1. It is now 4N content, okay? 2 of the blues and 2 of the reds, okay? The blue came from the father, red came from the mother. Now you have 2 from father's side and 2 from mother's side in this phase. Then at the end of meiosis 1, it will undergo, meiosis is almost like, meiosis 1 is almost like mitosis, other than little change here that I'll come to little later. The crossover and recombination will come back to that later. But then when we have meiosis 2, then there is no replication. Simply the 2 copies of the same chromosome, like say the mother's one or the father's one, they are taken apart. So now you have 4 different cells all having haploid content. And these are the germ cells. Okay. So if you think of human life cycle, we start with 1 sperm and 1 egg fusing together to make a diploid cell, which is called the zygote. Okay. And from there, the cell number increases and they become different from each other, and we are the full-grown human being. So every cell comes from that original diploid cell, the zygote. Now every cell is 2N. So if your germ cell also had 2 copies of each gene, that means if it was also 2N, then the next progeny will have 4N, right? It will keep on doubling. So, but that does not happen. So, from the diploid cells, a set of diploid cells are the progenitors for germ cells. They will give rise to germ cells. And there, those cells undergo meiotic division. Meiosis 1 followed by meiosis 2. And it is also called reductive division. So they have one end. So now whether you are a male, you have sperms. Each one of your sperm is haploid. If you are a female, you have eggs. Each one of your egg is haploid. So now when the new sperm and the new egg will combine to give rise to the zygote for the next generation, again it is 2n and it continues. Yeah? That's how the life cycle happens. Now this little thing is very, very important. You must have seen it, heard it colloquially within the family. Oh, this boy has these properties of his father, these characteristics of his mother. I do not know how many of it is based on solid data, but you have heard this, right? And if it is true, let's assume that is true. Then the explanation lies in here, that during meiosis, exchange of genetic material happen between homologous chromosomes. Okay, so a little part of father's DNA will be exchanged with little parts of mother's DNA. So if you look at it here in larger view, that this chromosome largely is from father, but this little part is from mother. This chromosome is largely from mother, but this little part is from father. So there is mixture of traits. This is how evolution happens. This is how, the next generation will inherit either, only the bad qualities of the father or only the good qualities of the father, etc. And, if the next generation is better fit for the society, then that is selected for. And in our parlance, we say survival of the fittest. So that particular organism is more fit to, give rise to the next generation. And then, that population will predominate in the crowd. Okay. So therefore, this exchange of genetic material between homologous chromosomes during meiosis 1 is a critical driver of evolution. This is not a directed process. This is random. It can happen from the middle of the chromosome, end of the chromosome, multiple places, all sorts of things happen. And most of these will be inconsequential, meaning it will not have any dramatic difference in the functioning or behavior of the next generation. Some of which will be detrimental. So that progeny or that kid, that next generation will not be able to survive. So the bad experiments are eliminated from the system. But there will be few that will provide selective advantage to the next generation. What do you mean by selective advantage? They will find it easier to find a mate. They will find it, they will perhaps have more reproductive ability. So they will predominate the population. And that will be now a slightly different, the whole population, if you think about, they will have a slightly different behavioral traits than the population that was a generation back. So these very slow changes happen, and that's how we adopt and adapt to changing circumstances. So it is very important that we understand this concept that at the end of meiosis, there are 4 different cells from one diploid cell, and each cell will have a haploid content, and there is a possibility of mixture of mother and father traits. Okay. I think I will end this lecture now. I will start lecture 3 with dominant and recessive mutations as lecture 3. Okay, thank you.